kits developed for particular variants developed. But I mean, the variants keep on changing so much that you have to evolve with that PCR target also like that. So if
Agreed.
you are detecting variant with the RT-PCR, so
If there is any
yeah,
specific kit that you have in mind, a design of a kit, not a kit, a
yeah.
design that you have in mind, which you
Mhm.
think will help us do a certain amount of variant analysis in wastewater,
Mhm.
and get a certain idea of what is happening, even in terms of a quantitative, not just qualitative, but a quantitative test. I
Mhm.
request you to please let us have a discussion. Dr. Mitali is here.
Sure,
Mitali has been
sure,
leading on the environmental surveillance front, and maybe we
sure.
can take that up. Dr. Mitali?
Hi, Lalit. Good morning and good morning. So yes, absolutely right. I think the experience and knowledge that you have gathered through the extensive research. I think the next end point should be, can we design a kit which can be commercially manufactured and
Yeah,
deployed for larger government programs, because that's the end goal. Yes, that will
yeah,
be wonderful, and we'll be happy to connect with you, Dr. Karina.
yeah, yeah. Dr. Mithali, I would like to draw your attention towards. I mean, I think we are not too late to start. I think Richa ji, can I say a few more things to
Yeah, just
Mithali ji?
Sure.
one thing
Yeah.
before we introduce you and then officially kick
Yeah.
off. Yes, please.
Yeah.
Sure, sure, sure. One thing I think in wastewater surveillance, one of the important aspects which CCAMP or IDX can focus on the processing of the sample.
Agreed.
Because the processing of sample, there is no kind of consensus for processing a sample globally. So if you can come up with a standard kind of a kit which is for processing the sample, I mean that is a bottleneck for all wastewater surveillance work.
Fantastic. I
Yeah,
think you hit the nail on the head, sir.
yeah,
Yes.
Fortunately,
yeah,
we have some work going on in that front.
right,
But you hit the nail on
Hmm.
the head. Even
yeah,
one more important thing is the cost of the extraction kit that
Hmm.
Promega or a QIAGEN will give you is
right,
so prohibitively expensive that
right.
the number of samples that you can do will automatically come down because of that cost prohibition.
Correct, correct, correct,
Yeah,
correct,
yeah. So
correct. And that's the
definitely,
strength of indigenous development. I really salute you
absolutely.
guys for this wonderful job you're doing. Yeah,
Thank you. Thank you.
yeah, yeah.
So, without further delay, and I know that a few more will join as we go ahead, very, very glad to have Dr. Khairnar back here with us. He is a medical microbiologist, public health, molecular biologist, accomplished across his graduation, post-graduation, PhD, and further work. And we do have a small Pondicherry connection, sir. I went to school there, but you went to something
Oh,
else altogether.
Zipmer, in Zipmer, yeah, yeah.
Yeah. But very glad because your research focuses on exactly what we've been talking about, which is molecular diagnostics of infectious diseases, specifically with an environmental and epidemiology focus. But currently, he is the head of environmental epidemiology and pandemic management at CSIR NERI, which is such an apt kind of an association for us, specifically because we have one vertical that entirely is working on technologies for environmental surveillance. And some of the associations that we've had include environmental surveillance approaches where he did talk to us before. He is part of the INSACOG, and now his lab has been recognized by WHO as at WHO's COVINET reference lab. And here he is today to talk to us about COVINET. I urge each of you to listen into this, because this is the current, it is almost like a status report on what's happening with COVID in a way. And also in terms of industries that are joining us, as well as my own team here, we need to look at it more carefully to see what exactly we can do to facilitate more work in this area. I mean, already in just, introductory banter that we've been having. Already two or three things came out, so I can only suggest that obviously the talk will elicit a great response. And thank you so much, Dr. Khairnar, for taking out the time. Dr. Khairnar, the stage is yours.
Yeah, thank you, Dr. Lalit. I'll now proceed with the presentation. Uh, yeah, Dr. Richa, can you confirm if the presentation is visible?
Yes, it's visible.
Yes. Yes.
Great, great, great. Yeah, so I'll be basically presenting on the two aspects. One is the WHO Coronavirus Network, and I'll be giving you a brief about what is this coronavirus network, and also how we can institutionalize the environmental surveillance. That is one of the impetus of WHO, that there are a lot of facilities created all over the world during the pandemic. So let's not let that go into waste, and try to use it in a way that we can institutionalize the environmental surveillance. And basically, the message is to keep the light on when we sleep. Yeah. So I am Dr. Krishna Khairnar, and I basically work at environmental epidemiology and pandemic management at CSI NEERI. We are recognized also as a COVIDNet reference and collaborating center for environmental surveillance. Yeah. So just taking you back to the days of 2020, when this pandemic was very new. That time WHO constituted a kind of a network of laboratories, and there were almost 26 countries across WHO regions. The main focus was about the kind of technical advisory group, which is tagged for viral evolution, and technical advisory group for COVID vaccine. So, in the due course, almost the 4 years passed, WHO realized that, there is a need to expand this network, because, a lot of aspects like animal health and environment and, kind of human health is interfacing, and there's a need to evolve this group into kind of which gave birth to COVIDNet. So the basic idea was basically, to have, kind of a coordinated effort, where we can not only look for SARS, but also for MERS, and in case there is any novel coronavirus. So the creation of WHO Coronavirus Network, as I said, One Health approach was the mainstay of that. And basically, WHO called for expression of interest when they were about to launch COVIDNet, and they had expression of interest received from many countries on human health focus, animal health focus, and also environment. And they basically, trickled down to kind of 27 reference laboratories and 7 consortia. For animal health, almost 3 FAO reference labs and 2 WOAH collaborating centers. And for environment, they are going ahead with the 4 WHO collaborating centers. One of them is CSI NEERI in India. And there is one consortium which is based in Singapore. I recently attended a meeting with them. So there are total 5 collaborating centers for environment with COVIDNet. So this is the map from WHO's surveillance kind of group, which basically shows the location of all these reference laboratories and collaborating centers across the globe. So this is the updated map of COVIDNet. And this was our first meeting. Richaji was asking in the morning that, so this is our group. It's a huge group. Basically, I can say that WHO commands the superpower because, under the ambit of WHO, almost the whole world is there. And it's absolutely an honor to be there with them. So 27th and 20th March 2024, we had a meeting. And basically, the meeting was to review the selection process and kind of consolidate the objectives, identification of key activities to support the core objectives, and development of a work plan, and definitely key performance indicators. So the EPI arm actually, as I was telling in the morning to Dr. Lalit, that, the basic message of this COVIDNet is to keep there is a steady decline in testing and sequencing. So kind of, to have a balancing act to this, declining trend of sequencing and testing, this kind of COVID EPI arm has been brought up where, kind of, there will be a coordinated effect, coordinated effort to basically, continue the surveillance and the testing in environmental samples for at least 20 countries, so that WHO has an kind of, its idea about what's going on around the globe, right? Rather than being completely oblivious to this kind of situation. Yeah. So, I mean, this is a very interesting slide. Basically, if I go to the left side of the slide, you will see the mosaic. And if I see from WHO's perspective, WHO is working with very diverse group and diverse kind of, diverse capabilities and diverse kind of, background if it is about WHO. So basically it is a mosaic, and you have to fit every aspect with its strength and weakness so that they can come up with a global plan. So I mean, we can say that crafting the mosaic and creating a framework for the globe. And if you look at the right side of the slide, you will see that at the bottom of the pyramid, reporting known and suspected novel coronavirus under IHR with a very basic thing, without which we can't proceed further. And almost 196, 195 countries are there on board, which is a very good thing. And for GISRS, which is a FluNet, which was the platform on which we all adapted to work on COVID-19, because the influenza platform was the most established platform. So on that, almost 130 countries are there in 2024. And then COVID-19 EMST and EGISR, which is actually kind of split between almost 71 countries are there for hospital, and 106 countries are for death in 2024. And then, of course, the wastewater and animal surveillance. This wastewater animal surveillance and this COVID EMST and EGISRS basically combined together gives us that One Health approach kind of strategy. And there are almost 24 to 40 countries in wastewater and animal surveillance working right now, respectively. And then the WHO SARI clinical surveillance network, which is basically going for a very specialized study in that 29 countries. So there are 20 countries involved in COVID right now, and basically, and there is an increasing number of component in collaborative surveillance. So I mean, this is basically one way to summarize, how the WHO is approaching towards this issue. So WHO has also created a dashboard featuring wastewater environmental surveillance. So this is one of the screenshot of the dashboard, and I mean, many are listening to this talk. Whosoever is working in wastewater or they have any data, they can definitely write to WHO, or they can write to me. They can copy me in the mail, and their data can also be showcased on the WHO portal. So we have a lot of data from different countries showcased on the WHO portal. I'll also present some of the data extracted from the WHO portal, just to give you an idea of what's going on across the globe. So the proposed COVID key performance indicators, like during the deliberation, there was a emphasis on integration with existing network, as I said, because a very solid platform has been formed across the globe. And then timelines of activities, to support the informed decision making, and accuracy of activities, which is a very mainstay of this complete campaign, because we had to be very sure about our detection and sequencing and reporting, and of course the capacity building indicators, because there is a need for certain labs and the regions across the globe where training material and surveillance, also right sizing the target is also very much required. So, I mean, these are the timelines, how the things went in WHO for the COVID, and I'll just skip that slide. So, coming to the, the variants which are, or the COVID-19 in wastewater surveillance, there has been moderate concentrations with increasing trend. If we focus on the Australia, the Perth, and if you see in the another region of Australia, which is Victoria, you will see there is a moderate concentration with increasing trends as per the data shared by them in the WHO platform. And this also, kind of correlates very well with the clinical data, the Australia, the Victoria data. And if you see this, this is a kind of a study from New Zealand, where you will see that they have tried to kind of strike a logic with wastewater surveillance on the kind of the burden of COVID-19 in clinical side. So I mean, that is the $1 million question, like how well or how best or how well gated you can actually take the information of wastewater surveillance to correlating it with what's happening in the clinical side, especially in the scenario when the COVID testing has almost gone very low. So in this scenario, how well wastewater can fill in those gaps and try to make, give the policymakers the ability to take informed decisions. So this is one, slide from the Gujarat, where this variant and all. You can see there is also, you can see there is there was a very high load, but it is going towards the trend is towards decreasing. Similarly, in Pune, Dr. Dharne is there, and in this also you will see that the data is still April 2024. More data is going to be updated soon, and this is also available at the WHO dashboard. And here also there was a very high load, but now the trends are towards decreasing, and there are actually the trends overall the trend across the globe is similar, but I have just summarized it like how it's going on across the globe. If you see what's happening till July 2024, so almost we have information from 27 countries in 5 WHO regions, and the data which I'm trying to summarize here is from early May to early July. And there is an increasing trend in 13 countries. There is also subcategory in that the high concentration continues to increase in New Zealand and Australia, especially in Perth. There is a trend of moderate concentration with increasing trend in Australia, Spain and Greece. And there is a low concentration with increasing trend in United States of America and European countries. And decreasing trend, there is a high concentration, but the trends are now decreasing. And in Canada and in Republic of Korea, there are low concentrations, decreasing trend. So, this is what, I am trying to summarize like what's happening globally. And there are only few countries who actually also show their variant information. Not all the countries are showing the variant distribution. So out of 21 countries, 11 countries are providing this data. And if you see, the KP variant is prevailing almost all the places. In 4 countries, KP2 is being reported, and in 3 countries, KP3. And, 5 countries have no details on JN.1 sublineage. So, we are awaiting them to populate that. Some countries have some limitation or inhibitions to share the variant data. So we are working with them, what are the limitation, and because variant information is very important in these days. So I'll just skip these slides and basically they summarize. So I will come to the institutionalizing the environmental surveillance research and capacity building for pandemic preparedness in India. This we are trying to show as a model, like how it can be as a COVINET collaborating center. We are trying to show how we can basically institutionalize this. So before going into that, I would like to briefly tell you about CSIR NEERI. CSIR NEERI has a very kind of old legacy, and 1958 it was established as a Central Public Health Engineering Research Institute. Very few people know about this fact. And we were basically a public health laboratory of central government, and we were basically working in a very big way on enteric viruses and that stuff. So at that time, in 1969, WHO Regional Reference Center for Air Pollution Monitoring, NEERI was recognized as that, and in 1972, environmental and clinical surveillance of enteric viruses was going on. It was continuing, and it had a WHO recognition also. And in 1974, basically after participation in United Nations Intergovernmental Conference on Human Environment, and this was briefed to then Prime Minister of India, Mrs. Indira Gandhi, and she herself visited NEERI, and she rechristened NEERI's name from CIFERI to so-called NEERI, National Environmental Engineering Research Institute. And then in 1975, health assessment of rural population of protozoas like Giardia and Entamoeba trichomonas was also going on as a WHO program. And then a dedicated virology cell was established in 1978. And, the scientists from virology cells in 1979 joined the scientific group meeting on human viruses in water and wastewater and soil. And in 1985, the surveillance of these are what I'm showing you. These are the highlights of the NEERI's kind of timelines in WHO, and they're trying to focus only on the relevant aspect. So in 1985, surveillance of bacterial and viral pathogen in drinking water with United States Environmental Protection Agency and CSR, there was a joint project. And then a novel method for concentration of viruses from water using bituminous coal method was developed by NEERI in 1988. Was from IIT Mumbai, and he was actually a part of WHO advisory group on environmental health. And in 1992, NEERI was designated as WHO Collaborating Center in Environmental Health. In 2011 itself, WHO was declared as a collaborating center for drinking water and sanitation. And in 2020, we became a regional hub of SARS-CoV-2 for molecular testing, basically the RT-PCR. And in 2022, we basically joined the INSACOG as a regional genome sequencing and central center for SARS-CoV-2. And in 2024, NEERI was designated as WHO Coordinate Reference Laboratory and Collaborating Center. So NEERI has a very big presence in India. I mean, NEERI is headquartered from where I'm speaking, which is CSI NEERI at Nagpur. But we have zonal centers in Kolkata, in Chennai, in Mumbai, and in Hyderabad. So, and almost our impact is on almost 73.42 million people of this country. So, just to give you a brief, what we did in COVID-19, we coordinated almost 2 million tests as a regional hub in this region of Nagpur. And, almost 3,000 plus SARS-CoV-2 genome sequences have been submitted in GISAID and IBDC, which is India's kind of platform portal. And we also came up with the novel non-invasive extraction-free gargle-based system for molecular testing and genome surveillance. And we also conducted the training for molecular testing and genome sequencing for healthcare workers and researchers across this region. So the CSR wastewater surveillance, basically there was a CSR wastewater surveillance also, which was lasting for a year, and from June 22 to May 23, very interesting data came, and this surveillance was so up to the mark that even many of the variants were reported even before being reported in the clinical samples. So I mean, Dr. Dhanne is also there, was one of the leads in this project with under the CSR network. So infrastructure wise, you see in the CSI and the CSI NEERI, I mean, only thing why I'm showing this slide is that, all these facilities are available even other places in India and also across the globe. So, I mean, the basic message that I want to give here is that we can basically leverage on the existing capabilities for, say, continuing the kind of monitoring for COVID or coronavirus in wastewater, but also we can leverage this for multi-pathogen detection. So that is one of the very important things which has come up in the WHO meeting, that leveraging on other pathogens also could be one of the important things in this. So, I mean, since I'm from India, and our dynamics are very different. If you go to Europe and Americas and all there, the dynamics are more of a urbanized kind of thing. But in terms of India, it is more of a rural, because 70% of our humanity is represented from the rural or health for all. Then we can't ignore our rural population. They definitely deserve better, healthcare support. So from that point of view, I have presented that, how we can do the wastewater surveillance in the rural area. And to be frank, right now there is nothing very clearly available with the rural surveillance, because there is one small category which says that, resource poor setup, but the definition of resource poor setup is not having sequencing or not having a molecular testing facility. But in India, the resource poor can also mean resource poor from the point of a proper wastewater network. So that is also a very important point in resource poor setting. So in India, these kind of when there is no wastewater, kind of a network, a proper wastewater network in major part of the India. So there basically we came up with these kind of propositions of, almost the data from government of India, like there are almost 1,66,000 open defecation free models which have been declared by the government of India. These can be kind of used for the wastewater surveillance in the rural area, and you will see that almost states have reported 1,198 functional sewage, the fecal sludge treatment plants, and 765 sewage treatment plants across 316 districts. So you may be very startled by these many numbers, but if I tell you the number of villages in India, it's almost 6 lakh plus. So there, then again, it looks that it is very less in proportion. So I mean, but still a lot of work to do. I mean, we can't. Also, there is a option of using open wastewater lagoons, which is a standard stay in the village and rural area, that can also be used for wastewater surveillance. So basically, the message is to decentralize the wastewater surveillance in a way that the neglected population or the resource poor population or the remote population of India can also be kind of roped in. Coming to the urban, the strategy is very clear, because our urban areas, the model of the WHO and the model of the urban sector very well fits here. And how NEERI is planning to do, we have actually tried to figure out how we'll kind of approach this, because it's a very big kind of, India is a very big country, and one lab can't handle all that. We would like to also kind of co-opt other labs also from India. That is also one of the important aspects of WHO. And we wish to initialize this with kind of metropolitan cities of Mumbai, Delhi, Kolkata, where the population is exceeding 4 million. And fortunately, we also have kind of regional labs or zonal centers there. They can help us in executing this. And the second tier plan is for the second plan is for the second tier city, where the population is ranging from 1 million to 4 million. And then towards the third city, we'll be progressing towards the third three-tier city, where population is ranging between 1 lakh to 1 million. So what would be the sampling frequency and the site selection? This is a very important thing which we all need to understand. Like if we talk about urban sampling, the wastewater sample will be collected thrice a week. Say you can distribute on alternate day, like Monday, Wednesday, and Friday. For rural sampling, in rural areas, the sample can be collected weekly. Example, you can fix a day for Monday or any single day, due to the lower population density, and also lower virus transmission rates as compared to the urban center. And this can definitely be evolved and deliberated further, but to start with something for which there is no clear kind of indications, we have deliberated on this, and we have come to a consensus that we will be doing it. And the sampling site selection, it is a very important thing, and it's one of the toughest job to do, and especially a country like India, which is very vast and very densely populated, and a detailed sewer shed studies needs to be conducted to identify critical junction, which can be a good representation of the sampling site. So I'm just giving you an example of how it was done for Nagpur. This is a 2-year-long effort at Nagpur, where the detailed sewer shed network has been completely mapped, and the Nagpur Municipal Corporation, as per Nagpur Corporation, the Nagpur is divided into 10 zones, and this is one of the inset of one of the zones of Nagpur, and you will see that how each node represents a kind of a manhole and a sewer network, which can definitely be used for a very effective wastewater surveillance. So like this, sewer shed networks in other cities or other areas is also very much required. And so the key consideration, basically, I will try to emphasize on the use case basis. You see, everywhere the COVID pandemic or the COVID epidemic or the COVID endemicity in today's scenario will keep on varying. So, I mean, it will be a kind of a use case basis. And here we would like to emphasize that, there has to be representativeness and equity. So, which I have already resonated in my earlier slide about a country like India, where it is more of rural than urban, and the catchment consideration, the choose for choosing a sampling point based on catchment size and alignment with the public health goals, and sampling protocol, which is a very important thing. Right now, we are working with WHO in coming up with standardized protocols or recommended protocols for wastewater also, and for other aspects also. So, I mean, also kind of, there are very basic things like, even the transport of the samples to the lab, a proper cold chain, because this virus is, if you talk about COVID, it's a RNA virus, and it is very fragile to degradations. So whatever are the kind of, takeaways of the clinical surveillance where the cold chain was very strictly followed. So the similar thing needs to be also be done for, environmental sample transport also. So, I mean, the early warning and sampling frequency, to detect any changes in the virus concentration, especially for high risk settings, sampling should be done daily, 2-3 times weekly. And for trend analysis frequency, like, that there is a variant and you need to analyze what's the trend, because we all have seen that, for example, if it is a Delta, during the Delta wave, the Delta only prevailed. And, we were always getting kind of, repetitive Delta in almost till that wave was gone. Similarly with JN, and you know what I'm saying. So, I mean, from that point of view, for monitoring significant changes over time, the frequency can vary from twice weekly to fortnightly. And for point of entry monitoring, because we are also working on airport surveillance now. So the airport, like those kind of point of entry, like airport and seaports, upon arrival of vessels from high risk area, this is typically done at the time of arrival. So it's at the discretion at the time of arrival. So I would also like to emphasize on the utility of the passive sampling in wastewater surveillance, because most of the labs across the globe are actually doing with the graph sampling method, including India. So we basically brainstorm on the utility of passive samplers. And passive samplers are basically very costly, especially those automated ones, with kind of automated samplers and all. But, we basically, deliberated on how we can have a cost effective and very cheap, passive sampler. It is not only cost effective for a country like India, but even for developed world, doing this in a big scale requires a lot of money. So, I mean, there has been a global concern on reducing the price of the sampling. So, we definitely support the passive sampling, and, basically the passive sampler with sorbent-based material, diffusion samplers can be a consideration, and are also the negatively charged membranes, which is not mentioned here, but I would like to add here. So the advantage would be, of course, as I said, cost-effective, ease of use, and also for long-term monitoring, because, the budget is always a limiting factor if you want to do long-term monitoring. So I would like to, share that, NEERI is deploying the in-house 3D printed Torbito samplers, which was basically, open sourced by BOSL.com in Australia. I'll share the photo of that also, which was 3D printed in our lab at NEERI. And, the data generated through this study will help in optimizing the design parameters of passive sampler as per the Indian condition, because this has not been done in India yet. We are the first lab going to do that, the passive sampler design in our lab. But on the design has been done by the BOSL.com in Australia. It is from the website. But the passive sampler was kind of 3D printed in our lab. So also we are planning to develop a drone-based deployment and retrieval system for passive sampler. This is under progress, and we are working very closely with our team, with Dr. Nikhil Lapsetwar and Dr. Piyush Kokate at NEERI. They are the engineers at our institution. So we may come up with that very soon. So this is the passive sampler designed by Dave McCarthy and the Monash University. The credit goes to them, and they have very, gladly made it as an open source. But this was 3D printed in our lab. And these files can be introduced into the 3D printer, and you can get these 3D printed. So, they look like Agni 5 and Agni 6 missiles, but don't mistake it for that. These are, kind of, plastic material. And if you open and see inside, you'll see that how the negatively charged membranes can be placed inside, and it can be just closed, and it can be just immersed into the wastewater for 7 hours to 24 hours based on your study design. So if any lab wants kind of, this kind of 3D printed sampler, can approach us, we can definitely, make and give it to them. We are also ready to handhold any lab for that. And the future prospects, so, so establish a continuous environmental monitoring of SARS-CoV-2 and also other viral and bacterial pathogen, which is basically, kind of multi-pathogen, leveraging on multi-pathogen. And as I said about the rural areas, we have access to wastewater lagoons and pit latrine surveillance system, which NEERI has been doing since ages in these areas. So I was the best person to basically propose that from India, being from NEERI, and this was very much kind of, appreciated at WHO, that, this can also be adopted in other parts of the world also, where there is, developing world. And of course, we need to have a kind of collaborative approach with animal and public health departments. If I'm speaking from NEERI, to strengthen the One Health mandate, and training of healthcare workers and researcher and environmental surveillance and pandemic management is also very important, which is also mandated for our lab. So my question here is that, basically, this I also resonated this question in my meeting at WHO, that most of the protocols and most of the SOPs available are all skewed towards urban setting. And, we, being from country like India, it is more of a ruralized than urbanized. So, for that, how far we can strike a balance, and how far we can draw a very, robust, kind of data and robust conclusion when we are working in the wastewater system. So a proper SOP for kind of, rural areas and the actual resource poor areas is very much required. So, yeah. So, we deliberated because we had a meeting in Singapore, also follow-up meeting, where there was emphasis on dengue and also chikungunya virus in wastewater. There were some groups interested in doing that. And also, as I said earlier, the leveraging for multi-pathogen surveillance would be also cost effective, because, there are certain, platforms like Twist platforms and all are available, where you can target the 3,000 pathogens. So I spoke to the Twist people. They said that you tell us what pathogens you want to target, we can give you a kind of customize that and give it to you. So, those kind of things are also available today. So, it depends on what kind of, design you want to have. And so thank you very much for your patience and time.
Thanks a lot, Dr. Krishna. This was fantastic. A lot of work has gone in, and it's wonderful to hear from you. Yeah, this is Richa, but I'm speaking from Dr. Mithali's account just to help us with the recording and all.
Right.
So anyhow, and you actually have touched upon so many aspects of, the pain points which had emerged during one of our surveys, which you too were a part of. If you remember, in 2023, we had conducted a survey just to understand what are the issues that people are facing across the country, specifically in environmental surveillance. And yes, you've touched upon several of those. And I did have a few people here who would want to, kind of carry over those conversations. But before that, we'll go to Dr. Mithali. And Mithali, you had a question for Dr. Kirnar?
Yes. So really very, very fascinating work, and how, although I think COVID was a difficult situation, but how so much of coherence and connections and has emerged from that, and truly can make a significant impact as and when we seem to see another pathogen of concern. So that's great. So my question would be, I think I have two questions. The first part is like you did talk about the sampling plan and did mention that like maybe the initial part of the work is through manual sampling, and you talked about 3 days, and depending, of course, that is flexible. Now, what is the time and uh, time of collection? And I think to some extent this question was answered when you showed the torpedo sampler, which is very well designed, and I think it can capture over a period of 7 hours, if I'm right. I heard you saying that.
Yeah.
So in that context, now how do you see we can talk to some of the indigenous manufacturers and get this design and fabricated in a commercial scale, so the cost will also come down? And have you adopted this in your current surveillance that is ongoing? So this is first part, and the second is like how you are interfacing with the municipal corporation, because finally, like in the long run, part of the routine surveillance probably needs to be institutionalized, as you said, within those bodies. So your thoughts, please.
Yeah, thank you so much, Mitali ji, for asking such a thought-provoking question. As I said in my presentation, most of the sampling modality deployed across the globe. I'm not saying about one lab or two lab. It is mostly graph sampling. So graph sampling has the disadvantage of just one shot sampling, and suppose if the wastewater is flowing and you are not capturing at that time, so it's very difficult to kind of find a time where you can do you will be the right person, right time at the right place. So that kind of thing is very difficult to happen with a graph sampling method. And but if you talk about the passive sampler, the range is from 7 hours to 24 hours. So you can keep it immersed, and you can let it get exposed to the wastewater flowing through, and you can have a much better kind of try at finding these targets in the wastewater than the graph sampling. And coming to your point of this, commercializing, it's a very fantastic proposition made from you. I think it is very much possible. These samplers are not only designed, tested, but also they have been checked with CFD, computational fluid dynamics, like how efficiently the fluid can flow through this and how efficient it is. So this is a design with a lot of work which has happened in the background. So I mean, this is open source, so I can talk to Dave McCarthy, and I don't think that he will have any issues with letting this, because we have already open sourced it. And you're right, the manufacturing capacities with the industry, like we are 3D printing, I'm not hiding it from you, but I can 3D print only one sampler one day. So, I mean, that is my limitation. So like that, from last 20 days, I have only 20 sampler right now, but it will keep on going. So, I mean, if we have an industry there, I mean, they can mass produce it, they can scale it up, and they can give it to, and these are very cheap. I mean, these are not even 150 rupees if you talk about the material, and the cost of making it is very cheap. So I think this is a very good proposition.
Is it reusable, or some filters will not be reusable, I think? Or
no, filters there which are there, you might have seen inserted as a negatively charged membranes and all. Those needs to be, because that will go into the buffer and it will be proceeding further. But the main body of the passive sampler can be reused, can be washed and can be reused. Yeah. And since it's a plastic material, it has durability also. Yeah. So, but I would suggest that better have more numbers, because it is cheap. So we can have a kind of a fresh piece there after 3 or 4 uses. I mean, let's not use it more than that, because the pores and all in that that get choked, and it's there is an issue of cleaning it effectively. So I mean, it's better to have more, because if you talk from US or UK point of view, it is available at the cost of a coffee. So I mean, for in India we can have a meal in that much of money, pura thali kha sakte, but still, I mean, considering the sensitivity of the test, better to have as a disposable ones. But if you have a limitation in the producing it, you can have it like you can use it for 2-3 times definitely.
No, thanks. And actually, maybe I think if Lalit is here, this is a great opportunity to bring this to a commercial level and have this instead of just replace the grab sampling with this passive sampling, because that will be a better mimic of the real situation, no? So yeah,
Yeah.
that's great. And the second was like, are we talking, because eventually some of the routine sampling or routine surveillance needs to be adopted through the health agencies and municipal corporations. So
Right.
something has to
Yeah,
move out from the
yeah.
research here.
Yeah, actually, I didn't answer your that question. What we did at Nagpur, that I can definitely say to you, and even with Pondicherry also, we did some work for there, not for COVID, but for some other aspect. But you need this, participation of municipalities, and in India the approach is mostly top down. I mean, if you go bottom up, you will get stuck somewhere, and you won't be able to move forward. So you need to engage people at the top. Like for example, I engaged my director. I actually, involved him into this kind of deliberation, and we had talk at the top level with the municipal commissioner and also the divisional commissioner, and they are very, I mean, kind of sensitive people. If you sensitize them and you make them understand, look, these are the advantages of what we are going to do, they are very happy to help you with that. And we got official letters from them. We got official permission from them to access all the wastewater network. And sometimes, if they are very kind of generous, they can even fund you with some money to do the work. But I mean, I don't expect much from them in terms of funding. I mean, we have many more other funding sources from where we can get, but their permission and their kind of facilities which are there, then their logistic is very important in this aspect. So I would always suggest that the approach should be top down, and wherever you have access, like you may kind of involve your the institute's top authorities, or also your, if personally, rapport is also very important in engaging them with like, for example, you can even have some local corporators or local MLCs into this. So it's kind of a people's job. So everybody is ready to come in, and especially in country like India. Yeah. So because in between I was in deliberation with WHO to give a letter like that to the Ministry of Urban Development or Ministry of Health for such kind of thing, but they were also kind of bit reserved in giving such kind of letters to them. So I mean, we had to come up with these kind of, because on ground realities are different, and we had to execute this study. So your question is very pertinent. I hope I've answered your question.
Yeah, no, fantastic. Thank you so much, and we'll be in touch surely. And I think we'll also touch, like with Lalit's inputs, this getting indigenous manufacturing, like I think is our goal, and we will take it forward. Yes.
Yeah, Mithali, this
Over
is
to
exactly,
Richa.
thank you so much, Mithali. This is exactly what I was going to, I raised my hand for the exact same. So, Dr. Krishna, there are two things that I would like to bring in right now. One is for the 3D printing, we do have one of the companies associated with the Index program called Bird Electronics, who has a 3D printer and can do it at a larger scale, can print this out, perhaps prototype it also.
Excellent.
With some more specifications if there are any, so they can help us there.
Yeah, manufacture actually, Richa, not just prototype, but manufacture.
Exactly.
Yeah. So what we can do, Richa, we can give them the file. We have a file which just goes into 3D printing and does 3D prints. So there is no need to optimize. There is no need to design. There is a file available. It's a Torbido file. It's an input file for 3D printer. They can just start producing it.
Wonderful. We'll get this thing connected. So we'll get you connected with them, with Mr. Anand there, and we'll definitely take this up. The other thing that I wanted to bring up right now is we do have a company called Nuome, who can kind of omit the need for cold chain sample, for sample delivery, a cold chain for sample delivery, So basically, they have a product called InstaPreserve, and we do have Dr. Elango from there, from Nuome, who can talk more about this product. And I believe I have spoken and I've brought up this InstaPreserve at one of the previous meetings also with the APSI consortium. So the basic concept is that, you just put your sample in. It's a gel-based sample collection tube. You put your sample in and it stays preserved. The nucleic acids and the protein also do not degrade over a long period of time, and they've done those stability assays. So this is one thing that I would, urge you to, I'll get you connected with Dr. Elango also. And I'd urge you to use it. And also, I think Dr. Mahesh Dharne was there when I brought this up during the APSI meeting. Dr. Lango, would you like to quickly tell us about it?
Yes, and a very good morning to everyone. Sorry, I got connected in the last moment.
Okay.
But the suppressor is basically developed for biobanking, storing the bio specimens at room temperature. We don't need any cold chain like dry ice or liquid nitrogen or deep freezers. We collect the bio specimens, blood, saliva, body tissues, and even that environmental samples also we have collected. The agriculture part also we have collected the soil, root, stem, leaf. This one point I wanted to hear, just to highlight is when me and Rajini, we went to a very remote tribal population near Metupalayam down Ooty and the background of a dam. We collected the soil samples, both in a normal way in the cover and also in suppressor. When we came back and we did the study on complete microbial identification with Oxford Nanopore next generation sequencing, the normal way collection with a simple plastic cover, we identified few organisms, but the one we collected with InstaPreserve, we could identify more organisms. And that one important thing is in the plastic leachate bacteria, where it is supposed to, it is in a very remote virgin land with a tribal population within a reserve forest. But how come a plastic leachate bacteria is present in that soil? Then we realized that Kunnur is a town below Ooty, is just 2 hills above this region, this reserve forest. When there is a rain, the water comes from Kunnur to this dam through this village. So that's how we get the environmental contamination from a town to the remote reserve forest area. That information we could capture only because we collected the sample in suppressor. The reason is that the sample which is preserved from the point of collection, which is very, very important. Otherwise, it leads to sample error. We collect the sample. By the time we get it to the lab and do the analysis, we lose some very, very important and critical information. This is what we realized when we did with InstaPreserve. So basically, this InstaPreserve is available in both gel and liquid format. Even now, we are in the hospitals, we are doing for histopathology. From the surgical theaters, when they are surgical procedure, when the tissue is collected, they keep it in InstaPreserve so that we don't have to use the formalin. It completely replacing the formalin, and now we are coming out with the publication also. There's another publication from Basavadaram Hospital in Hyderabad is also submitting by this week.
Great. Thank you so much, Dr.
Great,
Lango.
great, Dr. Ilango. And so when you are with Rajini, nothing is impossible. So yeah. So
Yes.
just from curiosity point of view, like for these kind of membranes, and suppose if I'm using even mood swabs, these are basically cotton swabs. So is it possible to transport them like cold chain free?
Exactly, you can do that. See, the thing is, we have been evaluating for the biological things, like we have done with the microbial and even gene expression, protein expression studies we have done, and we got good results. But some of them, like metabolites, if you ask, that we have not done. That is next level we are taking it up. Even the biochemical parameters. Suppose you collect a blood sample in InstaPreserve. Is it suitable for a simple CBC analysis of a biochemical parameter? That part we are now working on it.
Great,
so we can work out how fast we can customize it based on our need. We can work together. Absolutely, there is no problem.
Sure. Excellent. Yeah.
Great. Thank you so much, Dr. Lango. We will go to the next comment from the chat. We have Dr. Monica Nolan. Dr. Monica, would you like to talk about this? Would you like to share your experience?
Yeah, Monica Nolan, she's from WHO. Thank you, Monica, for joining. I welcome you. Yeah.
No, pleasure. Yeah, just very briefly, and this is really wearing my former hat. I used to lead the wastewater surveillance program for the state of Victoria in Australia, and we worked very closely with Professor David McCarthy and his team in terms of these sampling innovations. And I just wanted to say briefly that we had a very good experience implementing very widely passive sampling as part of our SARS-CoV-2 program. I think it's very important that for each specific pathogen, you need to have a validation process, thinking about how the pathogen associates and disassociates with the membrane. But Dave's team and lab did all of that work. So we had a lot of confidence in understanding the uptake, both of COVID as well as a fecal marker PMMOV, so we could interpret the quantitative results. And in our sampling, we have a reticulated sewage system. So we were implementing it at various levels through manholes as well as the wastewater treatment plant level, even down to building levels. And we were able to use it in terms of sampling just for routinely, sort of twice a week or weekly, depending on the feasibility of deployment and collection. But that basically meant that we had continuous sampling because you were retrieving the sample as you were deploying the next sample. So we had complete temporal coverage. And then if we had a detection, we had a low COVID rate early on. So if we had a detection, we actually deployed quite for a short period, so even for just a 15-minute period, because then we had 2 separate positive samples that were also able to help us interpret the epidemiology and what was happening locally. So we found it an extremely flexible tool. And, I think Dave McCarthy and his team is just brilliant. And any kind of, as Krishna had said, he's made it available in terms of open source. We did quite a bit of innovation too in improving the design for our needs. But I think if there were, deployment in other contexts other than reticulated sewage systems, there'd be also some opportunities to further refine the design. So I really encourage. I'm sure Dave would be very happy to join with the group if you wanted to have some further conversations as to what we learned and sort of support your use of this in your context. Thank you.
Yes, and see, for that question from Dr. Nilon, and this publication is in press, and it will. I think we can send you the copy of it. So this is on histopathology. With regard to the other microbial thing, that a paper, a manuscript we are writing, so we can send those details to you for your understanding.
Yeah, I think the cross conversation that's happening is important. I think, and this is really interesting that a single talk can lead to such multiple, collaborative opportunities. And like Dr. Mitali mentioned, we are focusing on, and one of our verticals deliverables is surveillance, environmental surveillance technologies. So I think we will take this up, take some of the ideas that have come through forward. Any other questions from anyone across or comments from anyone across? Otherwise, we're just at the cusp of time. So I thought one more. And there's a lot of activity on the chat, specifically asking for connects to Dr. Khairnar, which we will take through. Thanks so much, Dr. Monica, for coming over. And yes, that is something that we will definitely look up to. Maybe we will connect with you and find out. Sometimes in the whole wave of indigenization, maybe we get a little too inward focus. It's great to have you come in from where
Australia.
You kind of contribute, because we need to learn, and we need to kind of take that forward. Dr. Mitali, you had a question.
Actually, wonderful to hear from Dr. Monica and the insights she shared, and also drawing attention that you need to validate your pathogen panel, and especially with this approach of multi-pathogen surveillance. So yes, one has to validate the performance of it prior to deployment. But great to hear from you, Monica, and we would wish to be in touch for this whole indigenization effort.
So there were a couple of things that have emerged, multiple connects, like Lalit you mentioned. So just wanted to bring up that we have a medical doctor, Dr. Harjeet Singh Man from Gandhi Medical College at Bhopal, who is also into environmental surveillance, and he has requested for a connect, which I will facilitate in the coming week. Besides that, we have a couple of questions from Precision Health. Sirish, would you like to go ahead and take them? Bring up your questions?
Sure. Can you hear me?
Yes, loud and
clear.
Thank you, Dr. Mitali. Dr. Khaina, first of all, thank you for the great presentation, and really the work is really commendable. The question was on the stakeholder engagement. I mean, definitely, research community is definitely one very important stakeholder in the entire environmental surveillance business. But have you thought of engaging with the government? And you mentioned about the municipality and the district stakeholders there. But is there any sort of response you have got, especially in the safe period where you have not been, you have not witnessed any sort of early warning signal of a pathogen or a disease? Um, is there any particular response for the government to take, or to which we can expect from them in terms of making this more sustainable when the funding is, let's say, over? Is there any particular sustainable model we have thought of which we can implement? That's the one
Yeah,
question.
yeah. Actually, one thing is that in terms of sensitizing or warning, I mean, like the system in India and also globally is such that it's like reporting type. I mean, we if we the problem with COVID is that there is a issue of hoax and also chaos, and I mean building up on certain things. So I mean, both scientifically and also administratively, there is a lot of kind of careful approach towards this, because anything kind of published or anything displayed publicly can be taken up by media or can be taken up by any entity and can lead to some kind of unnecessarily kind of chaos in the society. So I mean the reporting is very, I mean, kind of streamlined, where we report to the authorities. For example, in Nagpur, we report to our health authorities, with the municipal commissioner, because they are the policy makers. They are the ones to call the shots. I mean, we can give them our findings, but ultimately they are the ones to call the shot. They have weekly meeting. There is a kind of, during the COVID pandemic time, there were like Corona war rooms where they can meet us or they can connect with us over the kind of video conference, and we can deliberate on that. So I mean, like one of the example I will give you, like how it, of course, wastewater scenario is going to unfold, because right now such thing has not happened in wastewater that something from this has led to some kind of immediate policy decision. It may happen. I'm optimistic about it, but from COVID pandemic point of view, I will say that one of our municipal commissioner, who is basically an administrative officer, he was of the opinion that we should imagine a scenario where we don't have to go for a complete lockdown. I mean, if we have early warning system, we have early information. Suppose, for example, Nagpur is divided into 10 zones. So if we have a reporting of a variant or a variant of concern as per WHO in Lakshmi Nagar zone, we can go to trace it, and at the maximum have a kind of a limited localized lockdown, rather than locking down the whole of the Nagpur or locking down whole of the Maharashtra or locking the whole of the country. So those kind of things have actually learned a lot from what has happened, and information is the key, and the timely information is the key, because if you kind of report something very late, I mean by the time it has already spread to many communities, there's no point in that also. So I mean, and as you asked that what we can do in kind of post funding scenario, the answer is that you can't do anything without funding, because see nothing, because you see funding from any source is the one which can let your lab running. Like for example, I showed you the slide of the facilities available in CSIR in NEERI. They are there. They are the capital investment, and they need not be bought every day or every year. They are there for good 4 or 5 years easily. So, I mean, any stakeholders can make use of this facility by just covering the running cost. So the cost of health is never appreciated. I mean, when people fall sick, when there are hospitalization, when there death, then people make chaos. But
Right.
but the importance of avoiding that, that cost is much cheaper than the cost which you ultimately pay by putting a lockdown or giving so much of trouble or kind of problems to the society. So I mean that needs to be realized by kind of the authorities, that look, whatever they are going to fund or make this running kind of a running cost. That's why WHO has also come to these kind of things, like where this clinical testing or or genome sequencing can't be done to that scale because
Right.
it's very costly. So these kind of wastewater-based surveillance can be one of the ways to keep our one eye open or while we sleep, or keep the lights open when we are asleep, rather than being completely oblivious.
Right.
So funding is necessary for, yeah. You can definitely approach CSR, corporate social responsibility. You can approach government. Government also have their funds. So you have to be a fund hunter, yeah, to
There's
let
one
this
more
go.
thing I'd like to add, that we have an APSI network. Sirish, you're aware of them, I hope, right? We have the
Yes,
APSI network. So APSI has engaged with the PR organization called Chase India. You can look them up, and we can help you get connected if needed, and maybe they can help you because they are the ones kind of advocating the need for environmental surveillance and making it a routine scenario in front of the government. So, they're taking it up to the government for facilitating any policy changes that can be brought in. So that's one thing. Maybe they can, connect with them, could help you with some funding options as well, if there are
Yeah.
anything that might be coming up in the near future. The
I
other,
was actually
yeah,
just one small thing. I was actually meaning to say that funding definitely is a very big thing, but how do we institutionalize it for the government, and they can actually budget it out in their own annual budget, and they can actually play a role there by themselves. Now that a lot of work has been done on wastewater-based epidemiology, how we can actually handhold the government institutions by telling them or guiding them in terms of what exactly can be done on disease surveillance, because there are different models which are being used to generate early warning signals and acting on it, and wastewater water surveillance can be a one more additional model in that respect. That's what I was actually meaning to.
okay.
Absolutely.
In that case, also, I believe a connect with Chase could help you. Chase
Yes.
India, you can look them up and let us know if you need a connect there. And they are advocating this exact same thing that you're saying. I mean, among multiple others, but for environmental surveillance, this is one of the things that they are trying to get done through the government. Great, Sirish. I think we'll take one last question from Mr. Amirul, or Dr. Amirul, rather. Do you have a question or a comment, Dr. Amirul?
Yeah, yeah. Hi, Dr. Krishna. Thank you for your
Hello.
for your talk. Yeah. So I just had a quick question. I think you had probably touched this upon on the earlier part of the call, and I think Dr. Lalit also had basically probably touched upon this as well. Is basically in terms of how to think of or kind of look at the design of a multiplex qPCR kit for for the detection and or let's say genotyping surveillance of variants as they evolve, right? Obviously, variants as they're certainly evolving. I mean, like that's something you probably just wanted to ask in terms of would you have any thoughts of that, or in terms
Yeah,
of
yeah. So it's a very good question. Actually, I mentioned that there are certain, kind of groups available who are into this multi-target detection. But I will correct you on genotyping. Genotyping can't be done. This is genotyping. You need to have sequencing done, because without sequencing, without having a look at the complete genome or a partial genome, you can't basically go for genotyping for evolving variants also. So to keep a watch on how the variant trend is changing, you need to have sequencing going on. But to suppose you want to monitor kind of a known variant which is prevailing in a particular scenario, like if we go back to our waves of COVID, I mean, there, if we know that, this kind of Delta variant is prevailing, so I mean, those things can be actually, designed, and there were examples where, certain RT-PCR kits for a particular variant, like I remember for BA.2, there it was one of the first RT-PCR based variant detection system developed. And then for Omicron also, there was a kit specifically developed for Omicron. But or even other viruses that they keep on, mutating, they keep on evolving, they keep on changing their variant trend. So it's very difficult to, have lock them in particular, RT-PCR based protocol. You may miss that. So that's why it's always smart to have a generic target in that, which will keep on showing you the COVID-19. And of course, if the variant panel can be made or built around it. So if suppose that is missing, you definitely have a idea that there is a COVID-19 is there, but a variant has changed. So you can kind of report that way that the particular variant has been shifted to a new one. That is also one of the smart way to go for it from diagnostic point of view.
Yeah. So just wanted to add here, Index is partner of APSI to enable indigenization, and completely agree with Dr. Karinar that you need a combination. And in this effort of making multiplex pathogen kits for wastewater surveillance, so in collaboration with GenePath, we already have a respiratory panel specially designed, which like for quantitative wastewater with the right normalization biomarker. So this has been done as a toolkit configuration, and it covers many of the respiratory pathogens, including the influenza A, B, and then RSV A, B, and various subtypes of influenza and avian influenza. So this actually currently is undergoing performance verification at APSI sites who have been into this area of surveillance. Similarly, we are planning another, or it's in progress, that's the vector surveillance kit for dengue and chikungunya, Zika, and then the dengue subtypes. So if anybody has interest, we will be happy to talk to you, and I think bring the manufacturer also, so that there is a wider know-how of these new kits which we are trying to develop and get them performance verified.
I think, yeah, the GenePath kit is definitely one step. But even the previous kit where we were looking at COVID, and then the fact that we're also looking at extraction means that we're thinking right on the right lines. So multiple things happening on the environmental surveillance front. And again, couldn't have been a better setting than have Dr. Karinar come and both inform us, give us a few new ideas and ratify some of the things that we're doing. So on three fronts, Dr. Karinar, thanks a lot for coming over and doing this. We should talk more often, and we should have you kind of take a look at what we're doing, listen into some of the people that are coming in. So I hope that this is a restart for that kind of activity for us, Dr. Karinar. But I know it's already 11:20 approaching. So I will thank everyone who's been on this call for joining in. I know it's a Saturday. Please do interact with us during the week also. And thanks a lot to Dr. Karinar once again. And thanks all for attending. Thank you very much.
Thank you. Thank you, everyone. Thank you.
Thanks.
Thank you, sir. Bye. Have a nice weekend, everyone.
Thank you.
Thank you so much.